will also pump hydrogen, but it will pump less hydrogen. The way to say it is this, is that for every NADH that comes into the electron transport chain, a concentration gradient is generated steep enough, so big enough, to drive the synthesis here of 3 ATPs. So it represents enough potential energy here to make 3 ATPs. The less steep this gradient is, right, the less energy there is. And in fact, if there's no gradient at all, what happens to this? It stops. It's not spinning. So we need a steeper gradient to make more. So under optimal conditions, NADH generates a gradient large enough to produce 3 ATP. FADH2 generates a gradient large enough as it spins through here to make 2 ATP. And that's that ATP synthesis. So there really is kind of an uncoupling, if you will, or we can have an uncoupling of the electron transport chain and ATP production. But it can only be uncoupled so far because eventually we'd reach a point where all the hydrogen would be up here and none down here, or not enough, and the whole process would slow. So it's almost a continual thing, right? We keep pumping, it keeps diffusing, we keep pumping, it keeps diffusing, and the pHs don't really change that much. But we're always keeping this gradient as such like that, come back and forth, back and forth, back and forth. Is that okay? All right. Now watch this. So let's not worry about this picture, it's good. You should, your textbook doesn't talk about those, but Google ATP synthase and look at the structure of that protein. It's really kind of cool to see with the little turbine and things like that. It's really kind of cool. All right, so here's the story. Now again, I have often asked on the test a question where this is a graphic one would produce to explain ATP production. The question would be something like this. Explain the production of ATP via oxidative phosphorylation, chemiosmosis, using a proton motive force, something like that. This is the answer. You need to start with energy in the form of high energy electrons of NADH. Show me how you get a concentration gradient of hydrogen and explain how that concentration of hydrogen is used to drive the synthesis of ATP. So this would never be a good enough answer, but this would be a diagram one would use, much like I did over here, to draw to support something that you're writing along the way, like annotations or whatever to make it complete. But here's what we got going on, right? We got NADH coming in. Now let's assume that it doesn't matter whether we're in any of the cells. We're just going to assume we're going to start with NADH, and where NADH brings the electrons in. NADH brings electrons into the electron transport chain. There are a variety of enzyme complexes that help facilitate these reactions. There's not just a single enzyme, there's a number of them. And within this enzyme complex, there are several redox reactions that are occurring. We're passing electron from here to here to here to here to here, until ultimately I pass it over to something else. And then this thing here, Q, carries it from that complex to this complex. Then I pass it here, 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 lots of redox reactions in here to this complex, until I give it to something else here, site C, right? And then that carries the electron from that enzyme complex to this one. Then I pass electrons here, 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 here through redox reaction, ultimately to oxygen to make water. And then we're done. Okay? In each one of these enzyme complexes, there are enzymes that catalyze the reactions of the redox reactions that are occurring, and there are proteins that are proton pumps. That is, they pump hydrogen ions. So here, if you look at what's happened, I'll just be kind of, again, there's a lot of redox reactions, a lot of redox reactions, and a lot of redox reactions, but let's just pretend there's one. NADH brings the electron in. NADH gets what? Did it lose an electron or gain an electron? It lost an electron. Loss of electrons is oxidation. So NADH gets oxidized to NAD. Something has to get reduced. What ultimately gets reduced? Q. Q stands for ubiquinone. So we could just abbreviate and say NADH oxidized, Q gets reduced, overall exergonic, pump a hydrogen. Pump a hydrogen from the matrix to the intermembrane space. Okay? Now where's the electron? In Q. Q is now going to give the electron ultimately to cytochrome C. So Q is going to lose the electron, means it's going to get what? Oxidized. Cytochrome C is going to get reduced, ultimately. As we pass this electron through this enzyme complex, those are redox reactions. Overall exergonic, yeah, pump hydrogen. There. Now where's the electron? Cytochrome C. Right? Cytochrome C's got the electron. What's it got to give it to? Oxygen to make water. So cytochrome C now has to get oxidized. Oxygen gets reduced to make water. As that electron passes through this enzyme complex again, lots of redox reactions, releasing energy. What are you going to pump? Hydrogen. Where's the electron at this point? Water. Where's the energy? And the potential energy here, where we now have a high concentration of hydrogen here, right? And a low concentration of hydrogen here. Yeah? Okay with that? And as long as we have this bad boy right here, which is ATP synthase, right? Hydrogens, as they're being pumped up here, are going to just fall back down here to the matrix. This process here is exergonic. We're going to use that to drive the synthesis of ADP and PI to ATP there. Now, if NADH comes in graphically, this is why this kind of is nice. It makes 1, 2, 3. There are 3 pumping attempts, if you will. It's alive, but whatever. There are 3 pumping attempts to pump hydrogen. So that's where we can kind of intuit that 3 ATP per NADH. That's not really true, but it's okay. Just say that. And why is it that FADH2 doesn't do it? It misses this one. It actually passes it directly to Q. And if it passes it directly to Q, then it misses this opportunity to pump. So it only gets 2 pumping attempts, right? So 2/3 of the amount of ATP that can produce, because per FADH, it's only going to contribute to the gradient 2/3 as much as NADH does. So you can only produce then 2/3 as much ATP from that. Okay? Is that okay? We're good with that? Cool. All right. And then that's how we get all this ATP.